So I blame my brother-in-law. He has a plow on the front of his truck and he took it off this week. I said, oh, you did it. So now it's gonna be, and sure enough, pretty smart. Okay, so we left off here. So we're focusing on the seedless vascular plants this week, which will not be on the exam today. Looking at here plum moss, brown pines, how they stay evergreen all winter long around here, and you can see them on the forest floor as the snow starts to melt. And so you can see those very typical strobili at the tips of their branches. It looks very different than the rest of the plant. And that's where they're clustering all their sporangia, where they're producing spores. And so remember, the sporophyte phase is the dominant phase in the seedless vascular, whereas the bryophytes is, what's the dominant phase of bryophytes? Gametophyte phase. All right, so a big difference there. And here, I think we left off here looking at the life cycle, where they were zooming in here again to the closely packed sporangium tissue where the spores are being formed inside. Right, so that's usually at the tips of the branches of the sporophyte, and then they go through meiosis and they get dispersed out and hopefully land somewhere suitable and start to grow into the gametophyte. So when that spore is germinating, gives rise to the gametophyte, and the gametophyte, in this case, you're seeing it's monoecious, one house for both the male and the female, Antheridium and Archegonium. And then free water necessary for the sperm to find the egg. And then once that egg gets fertilized, we call it a zygote. And then that's going to start growing into the sporophyte. And so the sporophyte is the diploid phase. It has the full complement of chromosomes. Diploid phase, okay, and the gametophyte is half the number. So this is from your book. This one is not from your book, but it has a little bit more text just for studying purposes. It's the same idea as what we just saw. So use that for study purposes. And then just looking at some visuals of how do they, the different species might look out in their native habitats. So here you can see the strobili are quite plump and very noticeable compared to the last image. Here you're seeing strobili of they call this a pendant design, where the leaves or the branches are drooping down a little bit, but if you still zoom into the tip, even though they're drooping down, you can see an obvious shift here in the morphology. So that's the strobilus, and that's where all the sporangia is starting to form and spores. So if you take one of these and cut it, lengthwise and look at it under the microscope, it would look something like this, where you see the sacs of tissue, the sporangium tissue, where you see all the spores inside, and you can see all the spores look identical. They're the same tiny size. They don't look different from each other. So we say they're homosporous. They're the same. Okay, we're going to see there's going to be other groups that don't do it quite like that. If you cut the stem in half and look at it, you'll see a central cylinder right smack in the stem, in the middle of the stem. That's called the protostele arrangement of xylem and phloem. The xylem, very obvious red, and then the phloem are the kind of the stripes in between. More delicate blue cells are the phloem. So that's just smack in the middle of the stem, so that's protostele arrangement. And did you see the resurrection plant in lab where you looked dead and then you put it into some water and it starts to unfurl and green up? It's quite amazing. And then you can let that dry out again. Like we keep the same one over and over. We just let it dry out again. And then next year we'll put it into water and it just rehydrates. It's amazing. So this one is a little weird in other ways too, in the sense that its spores now are heterosporous. And I'll show you a picture of that. What that means is they look different from each other and they'll give rise to a male gametophyte and a female gametophyte. So I'll get back to that in a minute. Oh, well, let me introduce these terms right now. So when we say they're heterosporous, then they look visually different and they're going to give rise to different gametophytes. And so the spores that are tiny, we call those the microspores. And think about like the difference between an egg and a sperm, the relative size of those two. Sperm are tiny. Microspores give rise to the male gametophyte that will be producing sperm. And then the other spores that are much bigger, we call those the megaspores because they're mega, they're big. And then when that lands somewhere and starts to germinate, grow, it will develop into the female gametophyte that will produce that big plump egg, mega egg. All right, so think about those, try to link those ideas in your head. So these terms aren't going away either. We're going to talk about microspores and megaspores in the next group. So please start doing flashcards for that as well, so you get familiar, comfortable. So heterosporous spores are visually different and they give rise to different gametophytes, a male gametophyte and a female gametophyte. So that's, let's look up here in the sporophyte phase. And you can see again the strobulus idea at the tip of the branch. And then if you zoom in a little bit, you can see how they zoomed in now to the strobulus, and you can see a big fat, where they say megasporangium, that all that solid green inside the megasporangium, that's a megaspore, big fat spore. All right, and then in the other sporangium tissue, you can see little tiny dots. So those are the microspores. Okay, so when those land, so they got to go through meiosis still, that's all the same. And then when those land, they're going to give rise to a female gametophyte and a male gametophyte. So you see the microspore and the megaspore. Okay, so the other idea, so we say, some people call it the male gametophyte and the female gametophyte. Some textbooks and some of the slides in lab, they might, instead of saying male gametophyte, they might say micro gametophyte. And then for the female gametophyte, they might say mega gametophyte. So again, linking the idea that micro, think small sperm, male. If you see a mega word in botany, it's almost always referring to the female egg, the female, the spore that gives rise to the female. So on this slide, this is not from your book, but it does have those words. Where is that? So right here, you can see the word mega gametophyte. So again, that's the female gametophyte, mega female. It even has EG in the word, almost the word egg, okay? And then everything else here, they're showing a nice difference in the drawing of megaspores versus microspores up on the upper left there. Okay, so those terms we're going to start seeing more again in the next group. So, and then in this group, they happen to do this as well. So here is a shining club moss in the baby stages of the sporophyte. So what's happened here is that the female gametophyte, the mega gametophyte, so there was an egg there, okay, and the egg got fertilized by sperm. And then once that happens, it starts to develop into the zygote and then the sporophyte, right? So you can see that it's in the very young stages. So when you zoom into the sporophyte, and you see those different spores, okay, the megaspore and the microspore. If you looked at that under the microscope for real, you would see something like this. Okay, so that's a strobulus. And in this slice of the slide or the specimen, you can see 3 sporangia that have microspores. So all those spores are microspores. And then the lower right, that sporangium has 3 huge megaspores inside. Okay, so once, if that gets released out and lands somewhere good, these huge megaspores will develop into the mega gametophyte, which is the female gametophyte. Okay, the little tiny ones, the microspores, give rise to the microgametophyte, which is the male gametophyte. Okay, So these ideas we're gonna keep seeing into the next group of plants. Here again, stem tissue, and you can see the vascular tissue is just smack in the middle of the stem, the xylem and the phloem. So this is a protostele-like arrangement. And then if you were gonna look at the shining club moss in its native habitat, it does look shiny. That's why they call it that. It has a very glossy appearance to the plant body. So very pretty, low growing to the ground or bark of trees, for example. Okay, and then this phylum also has the Isoetes or the quillworts. So phylum Lycophyta has several different groups. Isoetes or the quillworts, you can see why they call it the quillworts, because the sporophyte is spiked up like that again, kind of similar to the hornworts that we saw in the bryophytes. And then they don't have any strobili at the tips of their quills, so to speak. So you have to look at the base. They have to do it different. So they're at the base here. If you look at the base area, you can find that's where they tuck away their megaspores and their microspores in there. So a little different. And then you can see how the microspores are giving rise to the male or microgametophyte. Megaspore is much bigger, giving rise to the megagametophyte or female gametophyte. So egg and sperm getting produced, hopefully fertilization. And then you have the zygote, which starts to grow up into the sporophyte. Okay, so this is from your book, and then this one's from a different book, but I put it in because it has a little bit more words in it. So we can use this for studying as well. Quillworts don't look like a whole lot in their native habitat. They're easy to miss because they blend in and their quills often don't stand upright. They kind of bend over, so sometimes you can't see it very easily if you're trying to find them in their native habitat. Here's a really nice picture showing the full. So they tend to like aquatic areas along the edges, of the aquatic areas. Okay, so this is everything we've talked about so far. That's phylum Lycophyta. And so there's several different groups of plants that fall into that phylum. The next one, however, is very sparse in terms of who shows up in the next phylum. So the next phylum is Equisetophyta. Other resources may call it Sphenophyta. I don't like that term. I like the equi because that makes me think about horses. Common name is horsetail. So horsetail, an old-fashioned term that sometimes people will use is scouring rushes. I have a hard time saying that. Scouring rushes. And so that's because their stem is very abrasive. And you can use them, like if you're camping and you find some horsetails, you can grab some and use them like if you burn your dinner and your pots are all nasty, you can use that to actually scrape your pots and pans clean. So it's like a scouring pad. You can find them around here in town if you just look along ditches or rivers. Along the lakeshore bike path, I often see them, but you can find them in a more woodsy environment as well. But look where you can see kind of consistently damp soil. You'll tend to find them around here. They're very distinctive. They're easy to spot once you train your eye to them. But Equisetum is the only genus, as far as we know, that's the only one left. That's it. And so it's a very small phylum. In that regard, there's not a whole lot of living representatives left. And if you look at Equisetum, the current living Equisetum genus, those plants, and you track back to their ancestors, they really haven't changed much at all other than becoming smaller. They've become smaller and more slender. They used to be much larger. So they really haven't changed that much at all, except in their overall size. So a related species, genus is called Equisetites, and that one was, it's kind of like the big brother of the current one. It was huge. in terms of it looked more like a tree in the Carboniferous period. So kind of weird to think about because nowadays you only see these little horsetails that I see come up to about your knee. That's it. They don't get any bigger than that. So they're much smaller. So the sporophyte is what's dominating. That's what you see when you're out hiking around. Very unusual. They have some of their stems are very have obvious joints. I'll show you a picture. It looks like they're put together with what kind of a Lego block idea. And then the surface of the stem is very rough. That's where you get the silica has been deposited in those cell walls of the stem to help protect it. So it's very abrasive. If you were to use it as a scouring pad, you would have a good effort. The leaves don't even look like leaves. I'll show you a picture. They say they're scale-like. They're really small. They're super thin. They don't really do what a normal leaf does. They're just brown and they're like stuck to the stem. They don't really look like much of anything, but I'll show you a picture of that. And then if you look at the stem where these little scale-like leaves are located, they just attach all the way around in the same spot. So we say they're in a whorled pattern all the way around the stem. And again, they're very flat to the stem. They don't stick off the stem in any way. They have true roots and they have rhizomes, those underground stems. And so they can pop up a new sporophyte every spring and just like put up a new one every spring. So they look for them this spring when you're out hiking or biking around, you'll start seeing them. Okay, so the sporophyte, very obvious once you train your eye. So here you're looking at two different versions of the sporophyte, horsetail sporophyte. So these things on the left that look like, some people might say, oh, those look like pine needles, but those are just branches. That's the horsetail producing these branches, these short little branches. And you can see how it's all green. So it's all doing the photosynthesis. And then these other structures that you see that are very distinct from that, they're just these long stems. So here's a node region where you have those scale-like leaves attached as a whorled pattern. Okay, so very nondescript. They're not very green, so they don't really even do much photosynthesis. And then you can see at the end of these stems, you see they're really unusual strobili, very distinctive. All right, so the horsetails, what they tend to do is they have some of the sporophytes are devoted, like all the green ones here with these little branches coming off, everything's green. All of that, those sporophytes, they're busy doing photosynthesis. And then the other ones will pop up too when they're ready to produce spores and go through that phase of their life. And so they actually have two different looking sporophytes. One devoted for photosynthesis, the other one devoted for making strobili at the tip and spores there. So it's a little different. So if you were to crack open those or look at the strobilus more carefully and look at the spores, they're all homosporous. They all look identical. And they have the sporangium tissue is very unique. When you look at it under the microscope, it has this sausage-like shape to it. And then the spores often have those strips of tissue attached to them. And again, under the microscope, it's very distinctive. I'll show you some pictures. And so those elaters, those strips of tissue, we call those elaters. They help with spore dispersal because those strips of tissue will twist and move when they get wet and dry. That causes a twisting motion, and it helps disperse the spores. So the gametophyte, depending on which species of Equisetum you're looking at, the gametophyte can be tiny, 3 millimeters. Like you definitely need good eyesight for that. Up to 3 centimeters that you could easily see with the naked eye. And they're usually on the surface of the soil and photosynthetic. And it depends on the species again. Sometimes they're monoecious where they have both male and female, sperm and egg on one being produced, or it could just be a male. And then what they find out is that through lab experiments, if you have a gametophyte that has both male and female on it, what they tend to do is stagger when do they become mature. And so what tends to happen is the archegonia will develop first and be like the egg will be ready to go, but on that same gametophyte, the sperm aren't ready. And what that's doing is it's trying to promote that egg is like, I'll get sperm from another nearby gametophyte because my sperm aren't ready yet. And that's a good thing because you want that mixing up of genes from a different gametophyte. And so it's a good strategy to try to promote cross fertilization, get the sperm from a different individual's gametophyte nearby. And so we'll see when we get into flowers too, that flowers can do this too, where they can delay either the like when does the pollen get released versus when does the egg get made. And so there's some interesting strategies to promote cross fertilization. So this is one's from your book, and again, looking at they're showing nicely the different sporophytes where you have one devoted to photosynthesis, the one that's showing the branches, and then the other one is more, I just want to focus on getting a strobulus and getting all the sporangia going with the spores. You can see the very unusual strobulus structure, and then you can see how when they say sausage, what they're referring to here is these things here kind of hanging off the tip of the sporangium, kind of hang down. They look like a sausage. And then if you look a little closer, you can see that's just sporangium tissue with the spores inside. Sporocyte is the same thing. We just shortened it to spores. If you want to get really fancy, you see sporocyte, but I like short words, so spore. And then those spores still have to go through meiosis. And then here they're showing the elaters. Those aren't spiders, those are spores. And then the strips of tissue are attached to the spores. And so those will help disperse. And then you can see that you have, in this case, they drew a monoecious gametophyte. They're trying to show that one gametophyte has both the production site, the antheridium and archegonia for sperm and egg. And then once you get fertilization, and then that zygote will start to grow and develop into the spore. So horsetails, once you train your eye to them, they're very noticeable. And they're certainly around here in town. If you start looking for them in the spring here, you'll find them. This is from a different book, but again, for study purposes, whoops, I included that if that helps you, but it's saying basically the same thing. And then I added the relators on the spores, so they didn't do it in the other image. So, and then here, whoops, one wrong way. Here's a drawing of the strobulus, and then they zoomed in to one region there. And you can see all the sporangium hanging off like a bunch of sausages there. And then a real one in real life. So the leaves are actually these little things right here that are attached at the node. You can see a little bit down here as well. Those are the real leaves, but they're just paper thin, kind of useless. They're just not doing a whole lot. If you take this strobulus and cut it in half and look at it in cross section, you would see something like this. And so this is a strobulus, and so you can see those sausage-like sporangium tissue. And then inside all of those are the homosporous spores that are developing. So they can produce one strobulus, can produce a lot of spores. And so here they're showing the difference. So it depends on how mature the spore is. Sometimes you'll find the elaters wrapped pretty tightly around the spore. And then as they get closer to being released, they start to loosen up and start to unravel a little bit. And then as these elaters get wet and dry, they'll twist, and that helps actually physically move the spores, hopefully to a good habitat. So if you look at some spores under the microscope, you might see this, a mixture, where you can see some of the spores, they still have the elater tissue wrapped around them. It looks like stripes. You can see that, well, this one's pretty good. And there's some over here and here. And then some of the other ones, the elaters have unraveled off. And so you don't see that. So there's a mix here. And then if you're out hiking around and you see the sporophyte that's devoted to photosynthesis, you might see a patch. Often there's like a big patch of them. And very distinctive though, especially if you look at the jointed stems and you see that pattern. And then here, this is somewhere local where it's kind of a bog, not boggy, but consistently moist soil in this area. And you can see there's, you know, a mixture, it's a mixed hardwood forest, but all of this undergrowth here, that's all Equisetum. So they're all over the place here. Here's a stem. So because they tend to be in very consistently damp areas, they don't have a ton of xylem. Like they have some xylem, but it's not huge. It's kind of modest. And you see a lot of air gaps, like these big holes. Those are just kind of air gaps. And so, and then here on the very edge of the stem, you can see how it's ribbed. So if you were to pass your fingers around that, you would feel that ribbing with your fingers. It's very obvious. So that's where the silica is really reinforced in that area, is in those ribs areas. So you have a lot of silica in the cell wall. And so very unusual stem anatomy, a lot of air gaps. And so they're kind of hollow. A lot of the tissue in there is just air, or a lot of the stem area is air. Here, this would be a baby gametophyte that germinated. And so depending on the species, you probably wouldn't see this with the naked eye. So here, we won't ask you this question because it's too hard to tell, but if you're curious what these dark little blobs are here and here, those are the male, that's the antheridia starting to form, and you're getting some sperm production, but that's really hard to tell. So here is a drawing. They're trying to show that there was an egg here. So this was the archegonium. This is the archegonium, and there was an egg here, but it got fertilized. So now it's developing into a sporophyte. All right, so wherever the egg is, remember the eggs don't move. The sperm does, but wherever the egg is, it stays put. So if you see a sporophyte coming out, you know that that was where the archegonium and egg were located. Okay, so Equisetum, that's it. That's the only genus that we know of, unless someone can discover another species somewhere around the world. That's all we know of. Now let's go into the last phylum of the seedless vascular plants. Now we have a huge group in this phylum. So your book calls it Polypodiophyta, but other resources might call it Pterophyta or Pteridophyta. So what the book says, Polypodiophyta. Big long word. So these are the ferns, the true ferns we call them, not whisk ferns. Whisk ferns are not true ferns. So you'll see the phrase true ferns a lot, and that's what they're thinking about here. This is the largest group of plants that go into this phylum, even more so than gymnosperms. They're more successful than gymnosperms in terms of sheer numbers. So that's kind of, most people don't know that. You know, most people think gymnos and angios dominate the world, but it's not the case. The ferns are the largest group other than the angiosperms. So it would be angiosperms rank #1 in terms of number of species, then ferns, then gymnosperms. Okay, so very successful group. However, unfortunately, we don't see a ton around here of different species. And so if you really want to see their diversity and their full splendor, go tropical. Go somewhere tropical. 3/4 of the species are found there. We do have a lot of ferns up here, but not a great number of diversity. And then if you are in the tropics, look up onto the branches, the tree trunks. That's where ferns love to hang out. They're not hurting the trees. They're just using the trees as a way to get higher up so that they can get better access to sunlight. Really diverse in terms of where they live and then their body, like how they arrange their sporophyte, what's their morphology, really a lot of diversity. So for example, there's some species of ferns that have Their sporophyte leaves are 9 meters long, 9 meters long by 4.5 meters wide. That's humongous. I've never seen one myself with the naked eye, up close. Mauritia, that genus. So tree ferns, if you've seen tree ferns, they can get humongous leaves. And then that goes all the way down to some aquatic ferns have tiny little petite leaves. Azolla and Sylvinia, which I think you looked at in lab, tiny, tiny leaves, but they're ferns, they're true ferns. Here, probably the most common is the bracken fern, and the scientific name is Pteridium aquilinum. You don't have to memorize that, but bracken fern is the most common in terms of sheer number of individuals. We have a lot of bracken fern around here. So this one is Mauritia. It's kind of hard to see the scale, but this is the one that can be 9 meters long. Somewhere I saw a picture of an older model Volkswagen Beetle, with the rounded car body shape, and they had draped one of these Mauritia leaves across the hood and over the top of it and all the way to the back, and it stretched all the way from the front of the Volkswagen to the back. And I can't find that image anymore. If anyone sees that, if you could send it to me, I would like that image. I haven't seen it lately. Tree ferns, here you're seeing some images of beautiful tree ferns. So again, you got to go tropical for these. My one grad student, Scott, years ago, he and his fiance went off to the New Zealand and found all kinds of tree ferns there. And somehow some tree fern spores got into his pocket, carried them home. So for a while there, we had some fun ferns growing in the greenhouse. So he was very good at, oh, how did this get in my pocket? He was really good at that. So I miss him. So here's tree ferns. Beautiful, beautiful tree ferns. One of these days, I think this is a New Zealand or Australia picture. I got to get there. In fact, I think this might have been one that Scott took. And then here you see aquatic. This is Azolla, very petite, but really cute looking leaves. So aquatic, they can grow and float on the surface. On the left, that's Sylvinia. Their leaves are a little bit bigger than Azolla, but they're aquatic too, really cute leaves. This one's really fun. This is a staghorn fern. We used to have one in the greenhouse, but I think it died off. We had a cold snap where our heating system couldn't keep up with the frigid temperatures, and we lost some of our more tropical plants. So I don't think we have a staghorn anymore, unfortunately. But that's very unusual as well. It looks like, steak, the horns or antlers on a steak. So when you look at a fern frond, they're technically compound leaves. And really that's what I want you to know is, okay, they're compound leaves. And then the only other word on here that I'd like you to think about is pinna. So when you look at one of the leaflets, essentially, that whole thing is called a pinna. Other than that, I don't really care that you know the other stuff on here. So this is the, part of the sporophyte. So usually when you look at least the ferns around here, most of the sporophyte is just the fern frond. You don't see any like huge big stem like a tree fern. Okay, so when you look at the sporophyte around here, especially, it's mostly just the fronds, the leaves, and they tend to pop up year after year after year from underground rhizomes, or rhizomes, which are underground stem. And then it's so cute when they first come up in the spring here. I don't know if you've noticed that before, but they coil into a really tight coil called fiddleheads because they look like the end of a violin fiddle. If you look at the end of the wooden portion, it has that curled shape. And so that's where they got that nickname. And so those will be one of the first signs of spring around here. If you can spot fiddleheads, yeah, that's a good day because you're like, yes, spring is officially on its way if the ferns are starting to pop up. So these fiddleheads, they have this weird initial asymmetrical growth that causes the fern frond to coil up really tight in this coil formation. And maybe that helps them come through the remaining snow and the leaves that might have covered that area. So it's just a little easier to punch through. They hypothesize. But anyways, then once they punch through, you'll still see the fiddlehead design, and then they start to slowly shift over to a more symmetrical growth pattern so that the fern starts to unfurl all the way and you can see what it really looks like. One thing to be aware of is if you landscape with ferns, like in the house I used to live in, I had ferns all over the place because I love ferns, and I forgot. Really wasn't thinking about it that in the fall they die off. And so if you're landscaping with ferns, have something else nearby to kind of take over in terms of visual beauty, because it'll just be a bare patch in your landscape if you don't pay attention to that. So they do die back, usually every fall. But then in the spring, again, that rhizome underground is going to shoot up more fern fronds. Now, they don't have any kind of strobili. All up before, you know, especially like with the club mosses, we talked about all the strobili, the Echinacea have that. Here it's different. So ferns are kind of weird. They don't fit into that strobili pattern. Here are some fiddleheads. You can see how the fern fronds are just really nicely coiled up. You can, I mean, when you look at it, you can imagine that that does help them kind of punch through the layers of leaves. This is just a really nice visual of a stem tissue. Really nicely done. That was in a competition. But anyways, let's go back to the strobilus idea. No strobilus. So what do they do instead? They have a structure called sori. Sori is plural. Sorus is singular. So it's not, this is not a strobilus. That's why they gave it a different name. However, it's an area where you're going to find a cluster of sporangia. Just like, you know, strobilus has sporangia. The strobilus is all about it, making the sporangia there. Well, ferns, they do it a little differently. They have an area called sori, but again, it's a cluster of sporangia, and most of the time, all the spores look identical. There's a few different species that will be heterosporous, but most of them are homosporous. And if you're looking for these, you have to turn the fern frond upside down and look at the underside. And I remember the first time I did that when I was a young girl, and I saw all these, what I learned much later are sori, but to me they looked like a plant pathogen was attacking the fern. So I'm like, oh no, this fern is under attack. It's diseased. And then I realized much later, oh, those are the sori. That's actually a good thing. Because they look brown. There'll be like this brown powdery look to it. And so it kind of looks like fungal spores. And so I thought it was diseased. And then sometimes when you flip over the frond and you look for sori, they can be camouflaged because they'll get covered by some tissue that stays green. And so you have to kind of look closely. I'll show you some pictures of that. That tissue that kind of covers over and camouflages, they call it the indusium. And so they actually use that to classify ferns. They'll look, they'll say, how does this fern make its sori? Where are the sori on the leaf? Like what location? Are they on the edge of the leaf or in the center? And then they look for indusium. So all of that they use to classify and identify. That's why these terms are coming up, okay? So the location of the sori, like where are they positioned? I'll show you a picture of some differences. They actually use that. You look up a field guide, you're trying to identify an unknown fern, they'll start telling you, look for the sori, look for indusium, and all of that. So here's a picture just trying to get across that idea. So all these bumps, those are sori. And then in this case, there's a flap of tissue over it that stays pretty green. So that's the indusium. And then over here, you can see the sori are more along the margin of the edge of the leaf. And then here, they're more central to the leaf. So all of that varies. It just depends what species you're looking at, but it can be very characteristic. So if you were to zoom in to the sori, it's a bunch of sporangia in there. So I'll show you some pictures of that. But if you zoom into just one sporangium, it's very characteristic. You'll see this design pretty much across the board with all fern sporangium, where they have part of the sporangium tissue really reinforced really nicely, and then they'll have some more delicate cells here that they call the lip cells. And that's done on purpose because when the sporangium starts to dry out, you know, toward the end of You know, the spores are almost ready to go, then they go through meiosis. What happens is, as the meiosis is going on, all of this tissue starts to dry out, get drier and drier. And the lip cells, because of the drying action, it puts tension, and that causes the lip cells to break open. And so it kind of tears open the sporangium, and that can fling the spores. And they've tracked, like they've actually done the projectile measurements, and the spores can get flung quite a distance, considering how small they are. It's pretty impressive. So here, what you're looking at is a fern frond, and then this is the underside of the leaf. So here, this whole entire thing, so the whole entire thing that you see dangling down below, that is a sorus. Okay, that whole thing is called a sorus. That sorus is composed of clusters of sporangium. So all these individual sacs of tissue, that's all sporangia. All right, so a sorus has a bunch of sporangia in it. And then you can even see on this one where part of the sporangia is really nicely reinforced, and then the other part is very thin. So it, again, going back to this idea. Okay. So with the naked eye, if you're just taking a leaf and turning it over, you might see something like that. Here, the indusium is covering every single sorus. So there, you can see there's a bunch of sori on each leaflet. There's a lot. And so in each one of these is clusters of sporangia. So lots and lots of spores are going to be released. Here is a really nice image of fern sorus, where you can see a ton of sporangia, all clustered there. And each sporangium is going to be making all kinds of spores. So ferns can be very productive when it comes to making their spores and then flinging the spores out, and then hopefully those spores land somewhere suitable, and that will start to develop into the gametophyte. The gametophyte's really cute. It looks like a heart, the shape of a heart. I'll show you a picture. They tend to be photosynthetic, so they'll be right on the surface of the soil. It's hard to see if you were out hiking around. It'd be really hard to see because they're tiny. Usually, they're very, very small. And then they have these little rhizoids that come off that link them into the soil to keep them stable. And they usually are monoecious, which means both the Archegonia and Antheridia are going to be there on the same one. And it's usually short-lived. After the egg and sperm unite, then the sporophyte starts to grow, but the gametophyte tends to die. And so it's short-lived typically. Okay, so not much to the gametophyte, but they're cute looking. So here you can see a kind of a heart shape, the way it's drawn with some rhizoids coming off. And then here you can see the sporophyte. So you're right in the cleft of the heart, that's where the egg was. Okay, so that egg got fertilized, and now the sporophyte is growing from that position. The life cycle, this is from your book. So same basic life cycle as what we've been looking at. It's just that we don't have strobili in this group. We have the sori instead. And they're showing the nice cluster of sporangium with the unique shape so that they can tear open and fling their spores. And then here they're showing a heart-shaped gametophyte, where in the cleft of the heart, you have the Archegonia in the cleft, and then the Antheridia tend to be where the rhizoids are. Let me show you a different. This is not from your book, but if that helps you study. Here is a gametophyte. So again, you'd have a really hard time spotting this. It's just tiny, tiny. This is what it would look like under the microscope. So here you can more clearly see the heart shape to it. And so we wouldn't really ask, I'm not going to ask you anything about this one, but that dark clustering right there, that's all Archegonia with eggs inside. And then over here, this darker clustering toward the bottom, that's where the Antheridian sperm are getting produced. Okay, So real cute, but pretty short-lived. All right. Any last minute questions? Exam stuff. Feel free to ask, but otherwise we're done.